Hello students, I hope that previous videos of this chapter genetics was helpful to you. Now we have come to the very important part. Again I will say that is Gregor John Mendel's experiment on inheritance. Alright, so he was a scientist who experimented and has given certain variations and what are the outcomes of this experiments we will study in this. Alright, so he has done lot of experiments, so we will see here. So monohybrid cross, Mendel crossed the pure breeding varieties with the first taking only the one feature means any one. You can see here he has taken what is Pisum sativum is P. Now biological name, this is the botanical name of P. 1 mark question will come, and I have seen that so many times this question has come which children avoid generally, and it has come. I have seen that in the question paper that the botanical name of P is the Pisum sativum. That is why I have just highlighted it. So these were the certain characters, or you can say the features, on which the Mendels have conducted the experiment certain. So in monohybrid cross he has taken only one feature. What is that? Flower color, seed shape, pod color. Then pod shape, what is the shape of the pod? You can see that color is purple, dominant recessive is white, seed color yellow, green, round, wrinkled, green, yellow. Pod shape, how? This is, you can see, inflated, then constricted. Flower position, how the flower, what is the position of the flower? Is this axillary, then terminal, and plant height is tall or dwarf. Alright, so he has conducted the experiments, two type of experiments, monohybrid cross and dihybrid cross. So in this monohybrid cross he has taken only one feature, alright, carried out between the one pair of contrasting characters. Alright, so first we will study about the monohybrids cross. You can take a screenshot of this also, alright, and just take out a printout, and you can study easily. So in monohybrid cross, that is the same feature, pure tall and pure dwarf. When he has crossed, then in the F1 generation, first generation, this box is called the filal F-I-L-A-L, filal box. Okay, so what will happen here? Two, you can see pure, one is tall, and the other is hybrid tall, then again is hybrid tall, and the last one is dwarf. Why it is hybrid? Because it is a mixture of dominant and recessive. Here also the dominant and recessive, and then tall, and then dwarf. Alright, here it is pure tall. So what will be the phenotypic ratio? In the previous video we have seen that phenotypic ratio means what will be the observable characters, what you will observe. You will observe that, you can see here what you will observe, that 3, 1, 2, 3. Here dominant will express, here also T is capital, that means dominant will express, here also T is capital, so dominant will express. So the phenotypic ratio will be 3:1. Now what is the genotypic ratio? Genotypic ratio is 1, that is pure, this is TT, TT, this is pure, so that is why it is 1 is, this ratio is 1, and 2 ratio is because T, this is twice. I can write it like this, just a twice, so that is why it is like this. And or I will just write 1 T is capital and then other 1 T is capital, so 2 is like this, but here also the dominant will express, and 1 is again the 2 small t. So that is why I hope that you are understanding what I am trying to explain you. So that is why the genotypic ratio means the correct, when you see this box, then what you can understand, that when you see this, then you can see that the phenotypic ratio is 3:1, and but the genotypic ratio is 1:2:1. So what is the principle based on this above this breeding experiment? Alright, that each pair of the contrasting character depends upon the pair of the genes. What is the pairing? it depends upon the pairing. So I have already underlined the main keywords. Just in biology, just focus on the keywords. All right children, so very soon I am going to post you very important video where I will just explain you how to write the answers, how to make your answers effective and get full marks. All right, while you are doing section B. All right, then each individual carries the genes in the duplicate form, and an individual produce the gametes, the sex cells, which have only one member of the pair of the gene. So this is all depends upon these principles. All right, so now next, see, in this we have taken only one pair of contrasting characters. All right, now the next one was the dihybrid cross, where the two, it is carried between the two pair of contrasting characters. That means here you can see Mendel tried several combinations. So here round and yellow. So for round and yellow, what he has taken, capital R and small capital Y, and wrinkled and green, small r and small y. So here you can see when he has crossed all the round yellow seeds in this. So here what will be there, capital R, Y, Y. So when you will solve this, just multiply this with this, then again multiply this, then multiply this and multiply this. So as a result, what you will get, the phenotypic ratio will be 9:3:3:1. This is the phenotypic ratio, that is the observable characters. But the genotypic ratio is very complex. All right, it is just leave it like this. There will be no genotypic ratio, is still in the process. All right, the experiments are going on. So what are the two kinds of ratio of hybridization? When it is, see, F2 generation, when we are considering the F2 generation, this is just the sum up of what we have studied, the two types of ratios. Monohybrid ratio, phenotype is 3:1, genotype is 1:2:1, and dihybrid ratio is 9:3:3:1, and genotype ratio will be, if you want me to give you the genotype ratio, just comment me. I have just taken it out. The genotypic ratio will be 1:2. Just a second children, it will be 1:2:4:1:2:1:2:1:2:1. This is the dihybrid ratio. All right, sorry, this will be the genotypic ratio of the dihybrid ratio. All right, so it is very complex. It must be written in your books also. All right, so go through these important things, and this is very important topic because the questions are coming from this topic too. So I will tell you that this genetics chapter is very important. You cannot leave a single thing in this. You cannot leave any single topic, all the terms. All right, and the sex linked inheritance, then we have studied the characters, traits, all the new terms in this, and then Mendel's law, then what is the tongue roller. There will be so many questions will be coming in the picture based questions. I will be also discussing in those things. All right, so go through these videos, and if you have any doubt, then just comment me. Okay children, bye bye, God bless you, take care.